opportunity. So I guess if I can just share my slideshow, we can start.
Yep, it should be good.
Yep.
For you.
Yeah, I'm getting there here. All righty. All right, let me jump back. There we go. All right. Hopefully, is my screen sharing?
Yep.
It is.
Okay, great. All right. Well, thank you. So what I'm going to try to do today, I know they entitled this talk "Everything You Wanted to Know About COVID-19," but didn't know who to ask. Hopefully, I can be one person that you can ask questions to, and I can try to give you some answers and clarify some things, knowing that there's still a lot to learn and a lot for us to know, which is true really about all respiratory viruses. And I'm going to touch a little bit on that as we go along. What you're seeing here on my title slide is a picture from 1918. These are nurses in 1918. And then, of course, nurses today taking care of patients with this SARS-CoV-2 virus. And so in some ways, a lot of things haven't changed despite the advancement of technology. And it's actually interesting to see the parallels. But at the same time, there are things we can learn from our history with respiratory viruses. And so we'll touch on that as well. So the first thing I have to do is just a quick disclaimer. Anything that I say to you today is not the official position of the US government, the NIH, Francis Collins, Anthony Fauci, any other, CDC, any other government agency. What I'm going to do is try to give you data and my personal opinions as a physician scientist who's been working on respiratory viruses and pandemics for the past 15 years. As you heard, I'm the director of the LID Clinical Studies Unit. Just a little more information about what we do. Basically, I run translational clinical trials of human viral infections, primarily respiratory viruses. Influenza has been the major focus of my work, especially trying to prepare for pandemics and studying pandemics and understanding how we can prevent pandemics. I've run numerous clinical protocols to look at the natural history of respiratory viruses, the pathogenesis, including studies where I actually challenge healthy volunteers with influenza. I've probably given nearly 500 people influenza for the purposes of studying it and testing vaccines. So I have a lot of experience seeing people at various stages of respiratory viral illness. We also study zoonotic infection, how virus jump from animal to human, as it seems likely happened in this case, and really trying to understand correlates of protection, long-term immune response in order to make better vaccines and drugs and diagnostics. We've run a number of phase 1 and phase 2 trials in that area and are continuing to develop vaccines. I have no interests or connections to any company or vaccine manufacturer. I am going to mention a few in my talk, but I have no financial interest in any of these things. I'm simply mentioning them to you so you know about what's going on. So the first thing I wanted to bring up is, as you assess the data that's being thrown at you daily, whether that's from the news media, we all get emails, I'm sure, from numerous medical sort of online magazine sort of type websites that send you news. And even in these preprints and some of the journal articles that are coming out very quickly, is in the setting of a pandemic like this, information is coming fast and furious. And this quote that sometimes is attributed to Mark Twain, but it seems that it's potentially Jonathan Swift who actually came up with it, really is true. A lie can get halfway around the world before the truth can lace up its boots. And I can show you examples of where data is being interpreted very rapidly. It's spreading like wildfire. But if you really go back and take a look at it carefully and try to understand it in the context of how it was generated, and also in the context of respiratory viruses in general, it's not telling you the whole story. And so we have to be really careful during a pandemic not to overinterpret data as really is happening on a daily basis. So and we'll talk a little bit about an example of that later. It's really been a tough time to filter out what's true and what we really don't know. So I wouldn't be a good virologist if I didn't present you at least a little bit of basics about the virus itself. I'm sure we have people on the call who have, various levels of knowledge and understanding of this, but I'm just going to go over a few quick things so that we're all on the same page. The SARS-CoV-2 virus, which causes COVID-19, which is the disease that you're all concerned with, is a beta coronavirus. It's enveloped, positive sense, single-stranded, and it's an RNA virus. I didn't even write that on the slide, but that's very important. The fact that it's an RNA virus is really important because if you all remember from your molecular biology classes in school, RNA does not have an error checking mechanism when it replicates. Therefore, you have errors, which then act as mutations, being introduced every time the virus replicates. This is true for flu, for HIV, for coronaviruses. And what you see is when you look at coronavirus in a patient or a host, you're not just looking at one single virus. You're looking at a milieu of quasi-species, a big group of virions that are functioning together. And all of those virions are not genetically identical. There's lots of little mutations that are there. So you have groups in different percentages within a host. Now, those all are not different strains. They're just different quasi-species of what we would call the same strain. And right now, there still is only one single strain of SARS-CoV-2. You hear people calling things strains, but a true strain is when a virus has completely new properties compared to another strain of the virus. So they become antigenically distinct, or they behave in a completely unique way from a previous strain. That has not happened with SARS-CoV-2 yet, despite what you might read. There have been mutations that have been identified. There have been changes in the virus, but none of those changes have been proven to significantly change the virus enough to be called a true strain. So I would argue that there's still only one single strain. We know that bats are the natural host of this virus, but of course they can infect other animal species and of course humans. The beta coronaviruses themselves include lots of different viruses, including common cold viruses, HKU1, OC43, which your patients and all of us have had probably numerous times. And the way the virus typically infects human beings is through infections of human epithelium in the respiratory tract or the gut. The virus itself falls into a clade, or if you look at this branch of a phylogenetic tree over here on the right, that is in the same group with SARS, the original SARS virus. They're actually very similar viruses. There's 4 distinct sort of clades within the beta coronavirus family. MERS is in another group, and HKU1 OC43, those cold-inducing viruses, are in another. Then we have this 4th group here that so far has not infected humans, but we should be worried about that. And then there's a small group that's sort of unclassified. They don't fall into any of these groups. But for the most part, there's 4 groups within this beta coronavirus family that we need to pay attention to. And of course, SARS-CoV-2 up here with SARS-CoV. And then just a little bit further about the structure of the virus, you have the envelope. You have a few very important proteins on the surface. So the spike, which is in the form of a trimer. This is something we measure antibodies against frequently, and you'll hear about as you read vaccine trials or serology studies. There's also the nucleoprotein, which is internal to the virus, which some studies are looking for antibodies against as well, and the membrane itself. And of course, there could be many other antigens, and there's other epitopes within the spike, such as the receptor binding domain and others that people are doing tests to measure antibodies against. But that's the basic structure. So of course, COVID-19, which is the name of the disease caused by SARS-CoV-2, you're all, I'm sure, familiar with the symptoms of the disease. They can be both respiratory and non-respiratory symptoms. You can have GI symptoms such as nausea, vomiting, diarrhea, neurologic symptoms such as loss of taste and smell, headaches, systemic, fever, fatigue, body and muscle aches, and of course the respiratory symptoms, primarily cough, and then people can develop difficulty breathing or true respiratory distress. Of course, we see other symptoms as well in different people, but I'm presenting to you the most common that have been reported in the literature and are reported by the CDC as well. There's a huge spectrum of illness, anywhere from it basically doesn't cause any disease at all the way up to severe sepsis, respiratory distress, ARDS, and unclear, possibly rare long-term sequelae. I think that still really needs to be defined. We've seen lots of reports of different and various things, and it's hard to distinguish between what was caused by people being on a ventilator or some of the treatments they might have received versus what's really being caused by the virus. The things that are caused by the virus, how common are they really? When you have an enormous number of people getting infected and you see a small cluster of these cases, what does that really mean? And I think it's going to take a while before we fully understand that. So just keep that in mind when you see these reports of these long-term sequelae. We really don't know for sure what's true and what's not. I think it's going to take time for us to tease that out. What's been reported so far in the literature, and I'm referring mainly to a paper that was published in Nature Medicine recently, is that the median time to deterioration once a patient is diagnosed with COVID-19, if they're diagnosed before they've actually deteriorated, is about 8 to 10 days for those who move on to severe disease. We have a median time of about 5.8 days of incubation from the time that we think people are exposed. Of course, these are estimates. They're not going to be perfect. And about 7 days to symptom onset. So there seems to be about 1 to 2 days between the time someone would have a positive test to when they would actually have symptoms. And that's actually very similar to flu. We find that people have about 2 days of positivity with influenza before they develop symptoms, but the median incubation is much shorter. It's usually 24 to 48 hours versus 6 days. It looks like in general, people have a median of about 20 days of shedding as detected by the PCR tests that we're using currently. But there is a question of how infectious they are during that period of time. So far, cultures that have been done in the best studies that we have, which again are not perfect by any means, they can usually culture the virus for about 8 days. Beyond that, during that 20 days, they don't seem to be able to culture the virus. So, does that mean that a person's not infectious after that 8 days? Absolutely not. It's still possible. It just means that the samples that we're taking, we're not able to culture. And so it's hard to know exactly what that means. Even this median 20 days of shedding, this is based off the current clinical testing we're doing. As you know, there are lots of different tests. There are deep nasal pharyngeal swabs, there's spit tests, there's oral swabs, there's rapid tests. There's a whole host of different tests being used. Many of these tests have had limited validation and may have only been validated in one scenario. For example, you may have validated the test in people who have symptoms, but not done a full validation of the test in asymptomatics. Well, that makes a big difference because someone who's got symptoms you're expecting could be positive. So when you see a positive, that positive predictive value is better because you expected them to be positive in the first place. When you're now using that same test in people who are asymptomatic, your positive predictive value changes because you weren't expecting them to have a disease. And when they are positive, now you have a potentially higher rate of false positives unless you've done a significant amount of validation work. You also have scenarios where people are combining samples to do tests for large-scale screening like the Major League Baseball is doing. Again, have those tests been fully validated for that? I haven't seen all that data. There may be a FDA colleague who could answer some of those questions better than I, but I do question how much of that we're doing. We have to move quickly in the face of a pandemic, but we also have to be careful and think about that data. So I just raise these as things to keep in your mind as you see data. You really need to be thinking about what all this means. Whether or not it actually changes how you practice, it may not, but it's certainly something to think about as you look at the data that comes out. So of course, we're in the same place where we were in 1918 with the influenza pandemic, and we don't really have a treatment. So this is with a patriotic treatment bazaar in 1918. What you see here, it says medicine free if necessary, and what they were giving out, it might be hard to see, but what they were giving out here in this little sign by this gentleman with the hat is whiskey, brandy, or stout. So that was the best they were doing in 1918, and we don't have any specific treatment at this moment that really has significant efficacy for patients, particularly the ones who are very sick. And so we still have a long way to go in that arena as they did in 1918. Also, as in 1918, good nursing care is the key to survival. So in 1918, the number one reason why you survived this pandemic, and remember, this had at least 675,000 deaths in the US in 1918. There were 195,000 deaths in 1 month alone, in the month of October. So this was a real killer at that time. The number one reason why you survived, if you were in one of these makeshift hospitals, similar to the Javits Center today, or you were at home, was that you had someone to nurse you, someone to take care of you, someone who kept you warm, kept you hydrated, kept you clean, hopefully prevented you from getting a secondary bacterial pneumonia, give you nutrition. That's what kept you alive. That was the number one reason why you survived. And today, to some degree, it's no different. Supportive care is what we're offering people, and quality supportive care. And the higher quality that supportive care is, the more chance you have of surviving if you end up in an ICU. And you can look, there are different ICU fatality rates depending on where you are, who's taking care of you, how they're doing it. But the key is good quality nursing care, really taking care of people. And so just as a comparison with COVID-19, we have 139,659 deaths in the US. So we're not anywhere near 1918 levels yet, and hopefully we're not going to get there. Hopefully this will be over before that, and we'll have, maybe have some measures we can take. But just want to put that in perspective and point out this similarity. All right, so getting to it, and then this is very important, obviously, for this group, and you're all familiar with COVID-19 or SARS-CoV-2 infections and age-related death. And so I have a number of points I wanted to make. This was the most recent data as of yesterday from the CDC. Of course, these numbers change day by day, but this was as of yesterday. Of the deaths that they had recorded ages for, there were 121,374 deaths in the United States. 33% of those deaths were in 85-year-olds or older. 26% in 75 to 84, 21% in 65 to 74. This is a killer of people over 65. 85% of deaths in over 65-year-olds. That is where the majority of the deaths are happening. These are the people we have to worry about the most, which is often true in many scenarios, but is even more true now because for example, influenza, this is also true, but we also have down here at the bottom deaths. We're not seeing that with this. It's all up here over 65 almost. You see these much smaller numbers when you get down here into under 65. And of course, very important also in the young and the old are comorbidities. And the primary ones that we have to worry about are your patients with any kind of respiratory disease, diabetes, heart disease, and significant cancer. Those are the top ones. Of course, there's lots of others, but these are the top ones. And of course, these are the most common in the United States and really in the world. So these are big problems that our patients have that are putting them at risk of this death. And you do see, in fact, I've talked to people directly who tell me about their 95-year-old mother or aunt who, does dance classes every day in the nursing home, and she had COVID and was fine. They're not the ones we have to worry about. It's the ones with these underlying diseases, which is the majority of our patients, of course. Interestingly, with children, you have a very small, nearly non-existent death rate in healthy children. Really very little detectable disease because most of the disease in children is asymptomatic if it's there at all. We have not seen significant amounts of child transmission. Now, you're going to tell me that you read articles in the news today about a study out of Korea that refutes that. I'm going to talk about that in a second. Only 7% of all cases in the United States have been in children under 18. And if you look at the data coming out of the YMCAs, where they took care of 40,000 kids of frontline workers during the peak of the disease in New York, and you look at some of the foreign countries that did open schools back up while COVID was still circulating, there were 0 outbreaks associated with any of those childcare scenarios, and very little evidence of child to adult transmission in those scenarios. Now, the data I'm referring to that came up, of course, today after I prepared this presentation was a contact tracing study done in South Korea where they looked at about 60,000, I believe, families. They were testing them frequently, and they tried to identify an index case within the family. 11% of those 60,000 families were children between the ages of 10 and 18. Under 10, they didn't see a significant number of this, but between 10 and 18, they found index cases in that age group, or what they thought were index cases. And they report that 18.6% of the family members within those families developed COVID from what they call that index case. The problem here is, and some things that they didn't put in this paper, was they didn't talk exactly about the duration of time it took from what they're calling the index case to the actual case. Was it a week, 2 weeks? Was it 2 days? That makes a difference, because what we don't know is how did that child actually get the virus, given that during the time, and the authors of this paper report this, the schools were closed. If the schools were closed, it's likely these kids were home pretty much all the time while the adults were going out and doing the shopping and doing the necessary activities. Maybe some of them were working. So the disease was most likely brought into the home by an adult. So it seems more likely that there was an adult who was the index case. That index case was missed by the testing that they were doing. And it's possible that that index case could have transmitted not only to the child, but also to the adults in the household at the same time. And so it's very hard, based off the data that they present, to really make an argument that you know for sure that all 18% of those people within the families were infected by the child as the index case. And I think as epidemiologists look at the data further and really think it through, there's going to be a lot of questions about that data. So I really question that new study. I'm not saying it's wrong. I'm saying I question the data itself. I'm not sure. I think if we look at it very clearly and think about it from a respiratory virus perspective, there's a lot of questions we can raise. So personally, I know this was a question that everybody wonders about. Personally, from the evidence that we have here, I do think that schools being open is a reasonable approach. Now, that doesn't mean after a week, a month, a few months, you wouldn't need to close the schools because we see something different. But I think it would be very reasonable for someone to make a decision to open schools, see how it goes, and then change course if we start seeing a lot of cases or any outbreaks related to the schools. All right, getting a little bit more into the patients you take care of, nursing homes. This is data from the CMS system. So this is not complete data. They don't have reports from all over the country. They don't have full data sets from all over the country from every nursing home. What I'm showing to you is the data that they do have. They have 133,351 cases in nursing homes. You can see the distribution here in this map based on the color code where most of them are coming from. They have 83,476 suspected cases. So these are cases that they think were COVID, but they weren't able to diagnose either because a test wasn't done or they're just not sure, or there were multiple things going on and they don't know what actually caused the problem for the patient. And they have 37,213 deaths from those nursing home cases reported in the CMS system. And so this is showing you confirmed cases. The next slide, this is showing you where the deaths have taken place. So it's pretty much a similar map. Of course, more cases, more deaths. But this is very interesting. So you had 28% of the cases in nursing homes, people die from the cases reported. And more than 1/4 of all deaths in the United States have been in nursing homes. And that's probably underestimated because we don't have all of the data from all the nursing homes. So it's probably highly likely that it's really significantly more than 1/4 of all the deaths in the United States have come from patients living in facilities. That's a big number and really says a lot about the risk of these folks living in these facilities. So how do we prevent deaths from a pandemic? Well, the number one thing we can do is being prepared, which we could argue maybe we weren't as prepared as we should be. And not all of that is our fault, but I think there are lessons we can learn moving forward. And I haven't seen this discussed a lot, and I think it's very, very important. Improving the overall health of the population. I know that's all of our goals all the time. We need to do more. Keep people functional and out of these institutions as much as we can. And I heard a few people introduce themselves. They're trying to keep people at home, trying to take care of people. I know we all want to keep them functional as long as we can. That's really important. And reduce preventable diseases. When you look at these numbers, it's pretty sobering. I mean, we're in quarantine, right, for over 100,000 deaths from COVID, but we have over 600,000 deaths in the United States every year from heart disease. And there's a lot of things we could do to improve that. Look at the cancer numbers. Now, not all of those are preventable, but some are. Accidents, huge numbers, close to what we have in COVID deaths, and going on and on and on. You can see these numbers. These are big numbers. We need to lower these numbers. If we lower these numbers, we lower deaths from something like COVID, an influenza pandemic, you name it. This is really, really important. But obviously, it's too late right now for this pandemic. But for those people who haven't gotten sick yet, focusing on getting them as healthy as possible is really key. And for the future, this is going to be really, really key. So how else can we prevent it? Well, of course, there's lots of different topics and strategies being taken all over the country, all over the world, with masks, with social distancing. We're all familiar with lots of the controversy and arguments going on in both the political arena as well as the scientific arena. This is not something new. This discussion of how transmission occurs with respiratory viruses and how to best prevent it, to me, is a very old controversy in those of us who study respiratory viruses. And it's still not something we fully understand, even for the common cold or influenza or these viruses that we've been studying for 100 years. So trying to answer the questions about SARS-CoV-2 is very difficult. There are three main routes of transmission that those of us in the respiratory virus field discuss. Those are shown here in this sort of little quiz that I answered for you already. You have small particle aerosol. These are small particles capable of distant spread, usually smaller than 10 microns. They can get right down into the lung via airborne transmission. They can fly through the air. You can breathe them in. Then you have large particle droplet transmission. These are the larger droplets that usually come from the upper respiratory tract, whereas the small particles generally come from the lower respiratory tract. You typically have to have close contact with people for those droplets to fly close enough to you for you to either breathe them in or get in contact with them and infect yourself. And then you have self-inoculation, which is sort of similar. You're picking up these large droplets, usually off a fomite, an object, or maybe they land on you, and then you put them into contact with your epithelium somehow, either your mouth, your nose, your eyes, and so on. There is an argument. You will talk to different respiratory virus researchers and physicians, and you will find them argue for different aspects of this. There are people that say it's airborne transmission. That is the primary route. There's others that say it's droplet. There's others that say it's mostly self-inoculation. We can go on and on and on, and these people are everywhere. You can talk about this. My true belief is that all of these can play a role, but they may not all play equivalent roles depending on the virus and depending on the scenario. There's a lot of unknown variables here. Small particle aerosol transmission is probably most common in people doing high-risk procedures, intubation, bronchoscopy, maybe in environments where there's very significant singing, for example, in a choir practice in a small room. People are really, pushing out particles from their lungs. It's possible. I think in general behavior of our general lives, that's probably not as common as large particle droplet. Large particle droplets is what's going to be coming out of our nose and pharynx when we sneeze, when we cough, when we touch our nose and then touch things. So I think it's more likely that most of us are going to come into the large particle droplet, either inhaling those or self-inoculation. And I think those are the most common for most of us.
Dr. Memoli?
Yes.
I have a question that
Sure.
relates directly, that's kind of related to this. So I thought this is from Barbara LaRock, and she's asking, does rain impact this at all? Like if you have droplets in the air, you're outside and it's raining, does that like wash it to the ground?
Yeah, so there are a lot of different studies about environmental factors, particularly with influenza, not as much with coronaviruses. So, what I'm going to tell you is based more off what we know about influenza, but the droplet situation would be similar, is that humidity, temperature does play a role. Now, whether or not rain specifically itself would prevent or enhance the ability of the virus to transmit, I cannot give you that answer. But I can tell you that typically wetter environments, in terms of humidity, tend to be a little bit better. We believe that colder environments are a little bit better, but that's not necessarily held up with influenza, because we do have a lot of influenza transmission in the Caribbean sort of year round that happens in very humid, warm temperatures. Dry environments seem to be less beneficial to the virus, but a lot of this is still really, really unclear. And so we really just don't fully understand what plays a role in making these different sort of things happen. We still don't know the distances that you need to be away. I mean, we have this 6 feet rule, but that's not really backed up by any strong data to say 6 feet is the distance. Could be 5, it could be 10. You know, we don't know what the distance is, and it may be different depending on the person you're dealing with. If you have someone who's really coughing heavily, that distance may be longer than someone who has very few symptoms, right? And we also don't know the duration of time that you need to be in contact, or the amount of droplets or particles of virus that you need to be in contact with. This is all a mystery, even for influenza that we've been studying for 100 years. We don't fully understand this. We're just starting to try to get at this with some of the work that's being done in various places around the world.
We have several questions about the face mask.
Yeah, I was going to talk about those.
Yeah, okay.
Go ahead. You can go ahead and ask
later.
the question. You can go ahead and ask the questions, though, so I make sure I cover it.
Are face masks needed? And here is, that was one question. This is another question. Especially interested in knowing about a possible infection route through face barriers when interacting in the public. I think that's the face mask question as well. And then Ruth Junket asked about infections through the eyes. You talked about the nose and breathing it in, and should we be wearing protective glasses as well?
Yeah. So, okay. So let's talk about the masks. So when you look at these 3 routes of transmission that I put up here over on the right with this lady sneezing, when it comes to small particle aerosol airborne transmission, right? This is what you think of when you think of tuberculosis, which you all have, I'm sure, dealt with people with tuberculosis. When you deal with that kind of transmission, you have to wear an N95, okay? N95 is what, or stronger, right, is what protects you from airborne transmission. The surgical masks, the cloth masks, the gaiters, they have much less of an effect on airborne transmission. And if I was doing an intubation, I'm doing a bronchoscopy, I'm doing it in any kind of high risk environment like that, you really want to be wearing an N95, a PAPR, something that's designed to prevent airborne transmission, aerosol transmission. With the large particle droplets, now here's where the masks could be beneficial. And we use them. We've always used them when taking care of people with respiratory droplet precaution viruses. The masks, are they perfect? No, but they do can prevent some of the particles from getting into your mouth and nose. They also can prevent you from spreading some of your particles out. So they are functional as long as they are made correctly and are good quality. And depends on how much you've used them and all these different factors, which, you know, I think there are a lot of questions surrounding the quality of some of the masks that are available, where they're coming from, how they're made. When you get down to the cloth masks and the gaiters, they have all kinds of variation in terms of how good they are based on what they're made out of, how thick they are, and so on and so forth. And so we are in a situation where the masks that the general public are using are really of highly varying quality. And to me, that's a concern. And then on top of that, I think what one of the questions was getting at is, can some of the behavior surrounding the wearing of the masks actually negate any of the benefits of wearing the mask? So if you have people who are touching their masks constantly, which causes them to touch their face constantly, or the masks are getting contaminated and then they're touching their masks and then maybe touching their face or eating food or what have you, or maybe they're wearing their mask below the nose, you see this frequently, or the mask is very loose fitting, or you know, there's all kinds of things that you see people doing with these masks. could that negate the effect? I think that's a big concern, and we haven't done enough to study that, and we don't really, it's hard because we didn't have a lot of time to figure that out. And so, the true benefit of the masks, depending on who's using it and how they're using it, is going to be different, right? You using the mask to take care of your patient and a higher quality mask than your patient who's wearing a gaiter to do certain things is going to differ. But of course, risks are different for all these people as well. So, the true answer of do we need masks, how good are masks, I think is very hard to answer. I think it's clear that if people wear masks properly and are using reasonably good quality face coverings, it probably does reduce their risk of catching the virus if they come across someone who can transmit it. How much it reduces it, I don't know, and I don't think anybody does. And how many cases it prevents, I don't know, and I don't know that anybody does. But I think it's certainly reasonable if you're someone, particularly if you're someone at high risk, like most of your patients, that they wear the mask, but they should be educated on how to do it and make sure they're using something of reasonable good quality. The other thing I would bring up is you see masks a lot that are advertised with exhalation valves. You see them on TV all the time. I see people wearing them. These masks with the little valves that let you breathe out. That actually is defeating part of the purpose of the mask, because part of the purpose of the mask is if you're infected, to prevent you from spreading it to other people. Well, if you have an exhalation valve, that valve is actually sending it directly out to people, unless somehow that valve has a HEPA filter or something that really works, which I doubt most of them have. So just something else to keep in mind when you see these exhalation valves. So
how about the face shields or?
Yes, the eyes. Yeah, so the eyes, I think, definitely are potentially a route of inoculation. They certainly have been in flu. We know people have gotten flu through the eyes. I think it's possible. We don't know for sure if people have been infected through the eyes, but I think if you're doing anything of reasonably high risk, procedures of any kind with your patients, anything, you're a dentist, for example, anything where you're going to really be up close and personal with someone where they could, expel secretions into your face, I think it makes sense to wear an eye shield and would be very safe to do so that you're not getting those secretions in your eyes. So I would definitely say that's something you definitely should do as a medical practitioner. I don't think the general population needs to be walking around with eye shields, but I do think when you're doing anything close up and personal with folks, then it's reasonable to do that and probably should be done.
We are getting close to time, so could we, I want to give you like the 5-minute warning or
okay,
something.
yeah, no problem. I'm almost at the end here. I was just going to run through a few things about vaccines and antibodies, and then we'll stop and I can take questions for as long as you like.
Okay.
So just a few things about the vaccines. Vaccines are always a challenge for respiratory viruses. Of course, you're all familiar with how we develop vaccines, go through all these phases. We're on a very accelerated timeline right now with the COVID vaccines. But that also risks us not really getting the full data as we go through when we rush. The biggest problem is that the correlates of protection are unknown. And this is actually still true for a lot of these respiratory viruses. We still don't fully understand what it is about a person that you need to have in your immune system, in your immune response, that truly will protect you from disease. It's still why our influenza vaccines are still only 10 to 60% effective depending on the year. It typically takes many years to truly understand vaccine performance, and you may need a different vaccine for different types of individuals, like the elderly. One size may not fit all. There are lots of different approaches being taken, mRNA approaches like the Moderna vaccine that you've heard about recently. This encodes an antigen, and then the host machinery makes that antigen, and you have an antibody response. There's protein-based vaccines and vectored. Today, in fact, there was a phase 1 trial of the Oxford adenovirus vector vaccine that came out showing responses to the spike protein with an adenovirus vector. There's killed and inactivated whole virus vaccines. This is something that actually I'm working on. I'm not going to get into all of that today. There are virus-like particles. These are viruses that don't have RNA in them, so they can't replicate, but they look like viruses, pretty similar to the whole virus vaccine idea. And then there's novel proprietary approaches that are being taken, nanoparticles, replication-effective viruses, all kinds of things. To give you an example of the types of phase 1 data that we're seeing, This is phase 1 data from that Moderna vaccine that just came out recently. You can see they're looking at antibody responses after administration of the vaccine. And they did see pretty good antibody responses in the patients that received the vaccine, as well as some neutralizing titers. But this is in the serum. One big concern I have is that they didn't look at the respiratory mucosa. Is any of this antibody response translating to the respiratory mucosa, respiratory IgA and IgG? Because that's where the infection happens. Additionally, they did see a significant amount of adverse events, and this is something we have to keep in mind as we evaluate vaccines. So we'll see how things go with their phase 2 and phase 3, and hopefully they will have good results, as well as the Oxford vaccine. But I think there's still a long way to go in terms of really making sure we have a good vaccine. Now, what's the key to a good vaccine? Well, it's really understanding the correlates of protection. So which antigens are important to induce immunity? We still don't really know that. What types of immunity are important? Cellular, adaptive, I think probably all of them are important, especially when you're talking about elderly folks. And mucosal immunity is particularly important. And this is a problem because it's compartmentalized. What you see in the system, in the serum, in the blood is not always what you see at the mucosal level. You need mucosal dendritic cells, cellular immunity that migrate to lymph nodes to induce the adaptive immunity so that you have both B and T cell responses. And those often are resident T and B cells. The B and T cells within the lung, within the respiratory tree, within the gut are not necessarily going to respond the same way that systemic B and T cells respond. So again, only looking systemically for responses, to me, is a fault of many of these vaccine trials. They need to be looking at what is happening with their vaccines at the respiratory level where we want to prevent the disease. But of course, inducing respiratory immunity is very hard, as well as hard to measure the response because you have what's called tolerance. Your respiratory tree is subject to all kinds of antigens all the time. So it has a tolerance to those things. It doesn't easily respond. So it's harder to make vaccines that are going to give that good response. So the last thing I'll say is this is just a little quick couple of slides on the study that I'm doing right now. I'm leading a nationwide Serosurvey and blood sampling, which we're using home collection kits to take samples from people and determine how many people in the United States actually have antibody against the virus of different types, what levels they have, and hopefully, as we do this longitudinally over the next year, determine if these antibodies actually are offering any protection. We may not be able to determine that perfectly, but get an idea if that's true. And this is a small, rapidly deployed study that we started at the end of March, and now actually are finishing up the first phase, the baseline phase of sampling, and we'll hopefully have results coming in early September. I've just talked about these outcome measures. We don't have to get into it. But we've been recently funded to continue longitudinally and hopefully are going to start getting to the question of these correlates of protection that may help inform improving the vaccine. So we may get these first vaccines out, they may work, but they may not work as well as we need them to work. And so hopefully by doing these kinds of studies, we'll be able to inform how to improve them. And so we've had 416,000 volunteers actually for a 10,000 person study. And we'll probably end up enrolling about 12,000. We're looking for all subtypes of antibody against multiple antigens, and we're looking all over the country. So I'll stop there. I thank you for your attention. Baseball is starting up again, just as it did in 1918. And I'm happy to take questions and do the best I can in answering them.
Okay, I've got 2 questions from Betty Hinson. She says there are concerns about ACE inhibitors, the blood pressure medications on COVID. Is that harmful or helpful if you're on an ACE inhibitor? And
so, I think the jury's out on that. Obviously the ACE receptor is involved in these SARS-CoV-2 infections. And so it's just unclear at the moment whether that's playing a role, but it's certainly a concern. Hypertension has been a major risk factor of severe disease. But again, is that actually because a lot of those people were on ACE inhibitors or because they have heart disease, and that's a bad thing to have during a respiratory virus pandemic? So I think right now, I can't answer the question. All I can say is it's certainly something we should be thinking about and doing research on. But at the same time, we certainly don't want our patients to have out of control blood pressure. So it's a delicate thing to think about. I think if people are not on them and you have them controlled, then I probably wouldn't introduce them at the moment. But I don't know if that's been a really good drug for somebody. I don't know that I would take it off either.
Right.
Hey,
What about
Mario?
cholesterol drugs? Excuse me. This Betty Hinson also asked about the cholesterol drug being used as a possible treatment.
Yeah, so statins, for example, have been sort of promoted as antioxidants and treatments for a lot of different things. And there's sort of loose data that suggests that it may help with this infection or that infection, but there's still really no solid, clear evidence that it directly affects these viral infections in any way. So again, I think keep using them for people who need them for their cholesterol, but I wouldn't give them to anybody just for the purposes of trying to prevent severe disease or disease with coronavirus.
Okay.
Yeah. Hey, Mary Ellen,
Sure.
I figure we can go back and forth with some of the questions you received and the ones that came in. Bobby Behari says, how long can the virus stay in your system? I have an employee who was tested positive over a month ago. She has no symptoms, but continues to test positive.
Yeah, so this has been a big issue. So as I mentioned, the median shedding that was seen in that study that I mentioned earlier in Nature Medicine was 20 days by PCR. Now that's the median. So obviously there are people who shed longer. The problem we face is when we do PCR, we're detecting RNA. We're not necessarily detecting live virus, but we can't rule out live virus. So it's very hard to know what this long-term sort of detection means. On top of that, as I mentioned, we still don't truly know how good these tests are once people are asymptomatic and what the results mean. And so we're in a situation where we have potentially imperfect tests, already using a surrogate marker, which is the actual PCR of the RNA for the actual infectious virion. This gives us a scenario where we're have a difficult time interpreting it. For example, with flu, we also test by PCR, but we don't typically test people after they've been diagnosed. We just assume they're going to be negative after 5 or 7 days, and if their symptoms go away, we never test them again. So we don't have a lot of experience with this. We have very limited experience. So really understanding what it means is very hard. I think we're in a scenario where right now we're going to have people who test positive for a month, possibly even a month and a half, and we have to be very careful depending on what they're going to do. And I think, putting those people in contact with people at a nursing home, for example, or elderly people would be very risky because we just don't know what it means. So, the true length of time that this virus is infectious in the system, we don't know yet. We need to try to figure it out, but it's really hard to do.
Steve, it's 2:00. Should we ask people who want to hear the answer to other questions to stay on?
Oh, yeah. I would just, as long as the doctor is okay, are you okay staying?
Yeah, I'm fine. I can stay on plenty of time, no problem.
We're recording this, so we'll send a recording to everybody. But we do have a bunch of questions. Roz Cohen asks, will there be different vaccines with different dosages based on age, like the flu vaccine? Are seniors being included in the clinical trials?
Yeah, so, I can't tell you whether that will or won't happen. It's certainly possible. And it's even possible that there will be different vaccines for different types of people, as I mentioned. Right now, the phase 1/2 type studies that have been done have mostly been in young, healthy people. So we're generally talking about 18 to 45, because typically that's who you test out a vaccine for safety in initially. So we don't have a lot of data on elderly yet. As they move into phase 3, they will need to start including elderly people. And I hope we're going to see some of that data. But it's going to be very important that we do studies in the elderly and get an understanding. And so, I think it's reasonable to be concerned that some of these vaccines are going to work fairly well in the younger folks, the folks that actually have the least chance of death, and not work as well in that 80, that top level where we're getting the 80% of the deaths, and may have to do something different. And it's going to take time to figure that out. The good news is that hopefully by preventing spread in the younger people who are out and about, that could, obviously limit the amount of exposure our elderly do have. So it's not that a vaccine in young people won't have any benefit, but, I think it's reasonable to be thinking about all these things. So again, I'm hopeful that as we move through the summer into the fall, we'll start seeing the phase 3 data on the vaccines that are there, and we will get some data in the elderly, but I don't know how complete that data will be. And I think it's not going to be until we start using the vaccines in late phase 3, phase 4, that we're going to get the answer to that question. So I still think we're a long way off from knowing.
If you've got a question, Mary Ellen, I got plenty here, but
yeah, I know. This is one part by a long way off. What is any predictions on the length of COVID, or are we just going to learn to live with it?
Well, yeah. So there's different scenarios. I think the good news is that, as I mentioned earlier, this is a single-stranded virus and not segmented like influenza. Influenza as a segmented virus has the ability to recombine very frequently and change dramatically, which is why we have these shifts and drifts that occur yearly oftentimes and cause us to have to play catch up with the vaccine very frequently. This virus, as a single-stranded virus, won't be able to do as much recombining, and will also have some limitations in terms of mutations, and that as mutations occur, some of them actually don't allow the virus to replicate, and then it's harder for the virus to compensate. And without the ability to recombine, it has less of a chance to sort of compensate for those mutations. So I think that this virus will have less of a chance of mutating enough to avoid immunity to cause continuous outbreaks over and over again for years and years and years. Now, that doesn't mean it can't happen to some degree, but I'm saying from what I can tell and from what I think, I don't think that's as likely. I think it's more likely that this virus will circulate for some period of time. How long that is, I'm not sure. Could it be a year? Could it be 2? I think any and all of those are possible. And if it does mutate, it's likely that the mutations will cause the virus to cause less severe disease, not more. Because you have to remember, the virus's goal is to replicate, not to kill you. It would much prefer that everyone with the virus was walking around spreading it as much as possible, just from a natural selection standpoint. So typically, what is favored genetically in selection are going to be mutations that make these viruses cause less severe disease, not more. And that has always been true with flu. As a virus jumps from animal to human, the virus is typically, over time, from the time of initial pandemic to later, causes less severe disease. Some of that's a factor of immunity, but some of that is also a factor of changes in the virus. And I think it's likely we would see the same with this. So again, it's a complicated question. I think it's going to be with us for a while longer. Hopefully we're not going to see peaks that are the same over and over again. Hopefully what we're going to see is what we're seeing now, which is it's moving into states where they hadn't had their peak yet. And it may continue to do that for a period of time. Hopefully, we're not going to see it start re-peaking in places, like all of a sudden New York is peaking again. Hopefully that doesn't happen. But I can't predict for sure whether that will happen or not. And hopefully we'll have vaccines that will help us prevent that.
I've got one related question. I've only got 2 more questions in my stack, but I have one that's sort of related. So could I jump in with that?
Yeah, absolutely.
And we'll go back to our list. Okay. Identify what is a super spreader. Was that index person you were talking about? And
yeah, so an index case just means if you're looking at a group of people, it's the first person you actually diagnose with SARS-CoV-2. It doesn't mean there's anything special about them. It just means it's the first one you found. In the case that I was talking about, it doesn't even really mean that that's for sure the first person that had it. It just means the first person you detected in, which is why I question the data a bit. In terms of a super spreader, so what that is in terms of a respiratory virus would be a person who secretes a lot of virus for some reason. And whether that's through droplets or aerosol, whatever it is, it means they're making a lot of virus it's replicating significantly, and there's something about that person that's causing them to shed it out significantly, making it more likely that people around them would get sick. So for example, a person who's coughing a lot and has a lot of virus and, is going everywhere, coughing on everybody, you could argue is a super spreader. Whether or not there are people that are for that more likely to happen than others, again, this is sort of a controversial topic that we don't fully understand. We think that this sort of exists, but is it really a factor of the person themselves, or is it just a factor of where someone is in their disease or their behavior? This is all very, very complicated. So it just mainly means someone who is very likely to spread it to other people. And something along the lines of this topic that I forgot to mention in my slide that I really wanted to say is that, remember, the number one place where people have been getting SARS-CoV-2 is actually at home. It's in their house. And why is that? Is because close contact is the biggest risk. Spending a lot of time in close contact with someone is the biggest risk. So when someone has it and they go to someone's house and they spend a lot of time with you in your house doing activities where you're close together, that's where people are primarily getting the disease. Of course, it's different if you're in a nursing home, but it's basically where you live. I shouldn't say in your house, it's where you live. That is where you have the highest risk. So it's interesting because we're all staying home, and at the same time, that's the place where we have the highest risk. But obviously, those things aren't connected directly. It's just an interesting thing when you start thinking about it.
Let's see. Actually, this is a question from Ralph Sklar, who many studies have been aimed at detecting infections via reductions in heart rate variability. Recently, COVID-19 detection up to 3 days before overt symptoms have been reported. Do you have any information about the use of heart rate variability for COVID detection? And we had some folks on a few weeks ago with the Aura Ring who were involved with this study and the concept of, being able to predict a few days before flu and COVID occurs.
Yeah, so these kinds of sort of biomarker or biometric marker studies have really gained popularity over the past few years. There's something that, a lot of small devices and various kinds of wireless devices have been developed to do lots of different interesting, take lots of different interesting measurements from patients to try to study this. There's been a number of folks who've done significant animal work in this arena. And in fact, before all of this, I was planning an influenza challenge study where we were going to test out a huge number of devices in collaboration with computer scientists at University of Washington and Google to actually try to learn some of this. But of course, that study's on hold at the moment because of everything that's going on. So how predictive is a reduction in heart rate? I don't know. I think it's going to be very hard, especially with something like heart rate because of the variability. What we were actually going to do in our study was only look at heart rates during sleep to try to negate some of that variability that people are going to have based on their activities and things that they're doing. I think, is it possible that these things could be useful? Yes. Do I think we are there in any way, shape, or form? Absolutely not. I think we have an enormous amount of research that needs to be done to try to determine whether these kinds of biomarkers would really be valuable in predicting disease.
Diane Stern has a very practical question. Hi, is it safe to have rug installation done in my home? 2 men will be there for over 8 hours wearing N95 masks.
Yes, I think that as long as the carpet company assures you that the individuals they're sending to your home are not currently exhibiting any kind of concerning symptoms, and as long as you and no one in your family is actually exhibiting any significant symptoms, then I think it would be perfectly safe for them to come and install the carpets in your home. I think that, I wouldn't spend a lot of close, intimate contact with your with your installers, but I'm sure you weren't planning that anyway. But yeah, I would have no problem with people coming into my home and doing necessary work, as long as everyone is being honest about how they're feeling and how they're behaving.
Somewhat related, Steve. I lied. I had one more question.
Oh, no.
Someone asked about dentists too, or routine dental work. Should we go ahead? Should they go ahead with that?
Yeah, I think we can continue. I mean, I think that dentists and hygienists and so on are at a definite higher risk than other kinds of medical practitioners because of the close, intimate contact they're having with your nose and mouth, as well as some risk of probably aerosolized particles because you're keeping your mouth open and sort of breathing directly in and out that way. So I think for them, they definitely need to be wearing high quality masks and face shields. I would prefer N95s. I'm not sure what the dental associations are recommending at the moment, but that's what I would recommend. I think as a patient, your risk would be very low of catching something from your dentist. Again, as long as everyone in the dental office is being honest about how they feel and what's going on. And so as long as everyone's following standard procedure, I don't think there would be any downside. Obviously, it's important people can continue to get their dental work done. We certainly don't want an outbreak of endocarditis from people having problems with their teeth. So we definitely should continue to do that.
Let's see, a couple of questions regarding shedding. Can you define that? It's a term that I don't think a lot of us.
Sure. Yeah. So when we refer to shedding, what it basically means is we can detect virus. I would typically say using a high quality, hopefully approved clinical test for the virus itself. That's generally going to be a PCR. We do have some rapid testing, but generally when we're saying that, we're talking about PCR. So it means I take a sample from the patient. In case of COVID, it could be a nasal sample, it could be a nasopharyngeal swab, it could be a nasal wash, it could be an oral sample. There are lots of various ways it's being done. But I take that sample, I do a PCR, I detect the RNA of the virus. That's going to be called shedding. Now, true shedding would mean that there's actual virus particles, live virus particles that you're able to find in the nose, throat, lungs. But we're using the RNA, the PCR, as sort of a surrogate marker of that, which obviously has its limitations, as I mentioned earlier. But that's how I would define shedding.
Okay. Phil Golden has a very interesting question. What is a realistic apolitical timeline for an effective COVID-19 vaccine, in your opinion?
Yeah. So, it all depends on how you want to define effective. And so there's a lot of ways to define it, right? Are we looking for a vaccine that prevents people from dying? That's personally what I'm looking for. Are we looking for a vaccine that prevents people from getting sick at all? Are we looking for a vaccine that just reduces the level of symptoms? Are we looking for a vaccine that completely prevents you from getting the infection? Those are all different definitions. I think right now in the middle of a pandemic, the ideal scenario is let's try to keep people from dying. Can we knock down the disease to a level that is reasonable enough that we limit the number of people who end up in the hospital and end up dead. Now, if that actually prevents disease altogether, that's great, but that's really should be the goal, is reducing mortality. And then if we can reduce morbidity in the process, great. But in the long run, we may need to reduce morbidity as well, especially as we think about potential future coronaviruses that could emerge. In terms of timelines for all of this, if one of these initial vaccines, like the two I mentioned and some of the other ones that are a little farther along, pan out, meaning right now we know they can induce a systemic immune response. As I mentioned, we don't know if they induce a mucosal immune response, and we don't know if any of those immune responses that they're inducing are actually protective in any way. We're going to find out. We're going to find out when they go ahead and do late phase 2, phase 3. And hopefully we're going to find out sometime in August, September, October, how those are going. if those pan out, it's possible we could have something by the end of the year. Now, will that something be perfect? I would guess not. Is it sure to work in the elderly? It may not. How effective will it be? Who knows? It could be 20% effective, 50% effective, 30% effective, 90% effective. I don't know. It's going to be something we have to find out. And to some degree, it's going to be luck. Because as I said, we don't know the correlates of protection. If we knew, then we'd know what protection we were looking for, but we don't know. But they could also not work at all, or work very minimally, or only work in very limited types of people. And if that's the case, then we're looking at a longer process. And you know, could it be 2 years? Sure. Could it be 3? Could it be longer? Absolutely. And it may be a process where we have stages, where we have an initial vaccine that has some effect, works in certain kinds of people. Another year goes by, we improve on it, or we have another vaccine available. Another year goes by, we have something else. Now, whether or not coronavirus will even be a problem at that point, I don't know, but we still should work on it, because what we need to do, and we didn't do during SARS and MERS, is truly fully go through the development of this vaccine process so that we understand what the correlates of protection are and what the vaccines need to look like if we have another coronavirus emerge. And we all hope that doesn't happen, but we now know it can definitely happen. And so we need to be prepared. And so, and actually the vaccine strategy I'm working on is directly related to that. And so I'm looking to play the long game versus the short. So, you know, I think, yeah, you got to be careful. There's a lot of bluster and talking about we're rushing and doing this and that, but you know, it's possible we could have something quick, but it's also possible it could take a long time. And I always tell people the flu vaccine we currently have 10 to 60% effective depending on the year. The best we do when it's perfect match is 60. And that's not what you get in your patients. You get lower than that in your patients. Sometimes even with a perfect match, you only get 10 to 20% effectiveness in your patients. We're still, after, you know, 50 years, trying to improve on that vaccine. And we still have a long way to go to understand it. So this isn't something that just gets solved in a few months. This is going to take time to truly solve the coronavirus problem.
Yeah, really interesting stats there on the effectiveness of the flu vaccine. It helps put our expectations in check. Something, even though it's not necessarily related to older adults, but you had mentioned, you know, schools and kids and the transmission, but many of the teachers and adults are older. Any sort of thoughts on that? I mean, obviously there's potential for high risk transmission there, and it would be very important for the teachers to wear proper PPE.
Yeah, absolutely. So I think that, you know, we have data that shows that the amount of asymptomatic transmission from kids to adults seems to be low based on the data we have. Again, I don't know if that data is right. It's incomplete. It's not fully fleshed out. And, you know, the places where schools do open are going to be instructive in this. But from the data we have, it's reasonable to assume that that risk is fairly low, but it's not zero. I think if you are in charge of a school district or a school or, you know, a state, what have you, I think you certainly have to take each teacher and staff member on a case-by-case basis. There are going to be people working in that school that it's just too high risk for them to be there. And that's reasonable. They're going to have underlying diseases or be of an age where it's just too dangerous. It's not worth the risk. But there's also going to be children who are like that, right? Children with various kinds of immunodeficiencies, cancers, what have you. And so, you know, there are going to have to be things done and creative thinking to make it possible so those kids can get their education virtually and that those teachers can probably participate in that. But there are going to be many staff members who it is reasonable for them to be there as long as they're taking the proper precautions. And so I certainly think it's a completely reasonable approach for the teachers to be wearing whatever necessary protective equipment is deemed appropriate for the environment that they're in, and keeping as much distancing with the children as possible. So For example, my son is 4 years old. He goes to a preschool where, you know, the teachers are very close with the kids. They spend a lot of time with the kids. They do give the kids hugs and so on and so forth. When his school opens, you know, I think there's going to be a lot of less close contact, and that's sad for the 4-year-olds, but I think it's reasonable for the teachers to not have as much close contact with those kids as they typically would have had pre-COVID. So, but I think the data supports the idea that it is reasonable to open the schools. You're just going to have to look at each staff member and each child on a case-by-case basis to determine their individual risk, and of course, parent and teachers' comforts as well.
Another practical question that affects us all. Can you answer if groceries need to be washed when coming home after shopping? If so, what procedures should we follow? And I guess packages and things of that nature.
Yeah. So I think, you know, this risk, I think, was really greatly over-exaggerated at the beginning of this and really took hold. You know, I've seen some of the YouTube videos and so on. You know, the chance of picking up virus from an object in the grocery store or your mail is extremely low and probably very close to zero. You know, I think if your mail carrier has active COVID and directly coughed or sneezed all over your mail right before they put it in your mailbox and then you went and got it, okay, maybe. But the chances of that happening are slim to none. And the same thing goes for the grocery store. When you go pick out your oranges, unless the person who just was there a few minutes before you coughed all over those oranges, you know, these are RNA viruses. Obviously, they are enveloped, which stabilizes them, but they're not going to exist forever in that environment. They need hosts to survive generally. Sometimes they can survive in liquid for some period of time. We've seen that with flu. And yes, you will see studies where they show, oh, we found virus on surfaces after long periods of time. Most of those studies are done using PCR. It's not culturable infectious virus. And even if there is a small amount of live virus there, the likelihood that it's going to be enough to cause you an infection is very low. Now, is it reasonable to take certain precautions? Sure. What precautions would I take? Okay. I go to the grocery store. I'm not touching my face at all while I'm there. I'm getting what I need in the grocery store. I'm checking out. I'm coming home. I'm washing my hands before I start touching my face after I've, you know, put everything away and gotten rid of all the bags. Do you need to spray all the bags off with 409 or bleach? And I wouldn't go through all that. Washing your apples with soap, I wouldn't do any of that stuff. You know, you just take reasonable precautions, wash your hands. The virus is not going to hang out in your refrigerator for very long on any of that stuff, and it's probably not there in the first place. So, you know, if you see somebody sneezing on the fruit before you walk up to it, maybe skip that. But other than that, I wouldn't be overly concerned with that. That's not something I really worry a lot about.
If you're concerned about exposure, how many days should you wait before you test? And your thoughts on testing in general, like a couple of weeks ago, we had a senior living community that is doing weekly testing of all staff and residents. And it seems to be able to identify these asymptomatic cases and get them out of their system.
Yeah, so, okay, so the first question was how long to wait to test. So let me address that first. So as I mentioned, that median window seemed to be about 5.8 days. So if you're testing for actual virus, it's reasonable to wait 5 days to a week, I would say, if you're looking for somebody who was exposed and still may be asymptomatic. In terms of the testing asymptomatic individuals, as I mentioned, I have some concerns about that, although there are some environments where it's more reasonable to take the risk of false positives than others. And you just mentioned one, nursing homes. Obviously, we talked about nursing homes. More than a quarter of the deaths have occurred in nursing homes. We need to prevent the disease in nursing homes. Now, the one thing I would say is it's very important to find out what test is being used and has that test been well validated for asymptomatic testing. If it has, great. If it hasn't, you want to try to find one that has. And when I say validated, it doesn't mean used a lot for asymptomatic testing. It means they took a lot of positive controls, negative controls, and worked this out statistically. And there's ways that this can be done. In terms of, so, I mean, personally, I'm not a big fan of asymptomatic testing. That's just not something I really am a big fan of, but I completely understand why nursing homes are doing it. And I know some places are doing just all the staff, some places are doing the residents and the staff. I think as long as you're, as I mentioned, making sure you're using a high quality test and really carefully reviewing your data, then it's perfectly reasonable. And obviously, to be honest, it's the one scenario where I think it does make some sense to do. Whereas I'm not sure Major League Baseball needs to be doing it. But anyway, none of this is perfect because we just don't know how good all the testing is.
I'm trying to, we're coming up on another half hour here, so I'm trying to kind of just wrap up with some of the questions that are on topics that we haven't really touched on. Bobby Bahari has a good question. Would you recommend hosting flu clinics during the pandemic?
So yeah, I assume he means flu vaccine clinics. Is that what he's referring to?
Yeah.
So I've said this to a lot of people at NIH because a lot of my flu research has actually gotten held up because of all of this. And I say, we don't want to survive the COVID pandemic to die of a flu pandemic. So I think we do have to continue to give flu vaccines, and we have to find a way to do that, as well as the other vaccinations, right? I'm sure you give Pneumovax to many people per year. All of these vaccines are very important to continue to give people. They're going to help prevent COVID deaths because people do die with COVID and flu together. People do die of COVID and pneumonia. These things are all happening. And so we need to continue to do that. So I think you just have to be thinking about how you do it and, limiting the number of people who are together. And so, typically you might really pack them in for these clinics. You may have to space them out more, do them over longer periods of time, doing them in different environments. I think you have to get creative and think about it. So I don't have a perfect answer for you, but I do think you have to find ways to continue to give out the vaccines.
And a few questions came in on testing, but like testing protocols if you had surgery that was scheduled. But Allison just wanted you to reiterate a little bit why you're not necessarily a fan of asymptomatic testing.
I'm not a huge fan of asymptomatic testing because I'm worried that you end up with a lot of, so it all is dependent upon the test itself, the quality of the test that's being used. If the test hasn't been validated for that, then you're going to get a lot of misleading results. And that could end up having a very negative effect. And so it all depends on if the benefit you're getting out of it outweighs the risk. So if you're doing this in an environment where the risk of severe disease and death is very low, but the risk of having false positives takes people out of commission. So let's say, I don't know if this is a perfect example, but let's say you're in a pediatric clinic with mostly healthy children, and you're testing your nurses, and the nurses are coming out positive, and now you don't have enough nurses, and kids are getting sick, and you don't have people to take care of these kids, and the kids are getting sick with other things because other diseases are still happening, right? Maybe the risk of that doesn't outweigh the benefit, because even if you have one asymptomatic nurse, the chances that any of those kids are really going to get very sick and die are low. I don't know if that's the best example. I need to think about this a little bit more. But whereas in a nursing home, if you did have a transmission event from an asymptomatic nurse, say, to a resident, that could obviously be devastating to the entire nursing home. So in that case, it makes a little more sense. And if you take a few nurses out of commission because of false positives, it may be worth that risk. So I think it all depends on the scenario and the quality of the test that you're using and how it's being used and how the testing is being done. The other thing I worry about is a false sense of security, and that false negatives could actually lull you into feeling confident that you don't have the disease present. But you have to remember that whatever sample you're taking is only sampling where you sampled, in the mouth, in the nose, in the nasopharynx. It doesn't necessarily tell you for sure that the virus isn't present in the person's lung, in the nose, in the throat, somewhere else. And so we don't want to be lulled into saying, well, I've tested my whole facility and everyone's negative. We can just relax and not worry too much about making sure we're following all the procedures. We have to be vigilant about infection control, no matter what, whether we're testing or not. So it's just, we just have to be careful about how we're doing it and make sure we're getting value out of it, and that the money and time and the efforts are worth it compared to other things we could be doing.
Great.
Can I ask sort of a global question? John Hinkle, this is my last one. Ask about characterizing this with the differences in the deaths between the 1918 flu epidemic and this 1918 flu pandemic and this event. Would you just characterize this as a bad flu season?
So yeah, okay. So we don't know. Okay, so let me say a couple things, and I'll comment on that last statement. So a big difference between 1918 and every other respiratory virus pandemic in the modern era that we have recorded. So I can't tell you about what happened in ancient Egypt when they had a flu pandemic, but I can tell you about since 1918. There were no antibiotics available in 1918. An enormous number of people, probably more than 90%, died with a bacterial pneumonia on top of their flu. You and I both know that everyone who comes into the hospital with COVID pneumonia is getting some whopping combination of broad spectrum antibiotics, right? Vanc and Zosyn, something like this, especially if they have severe disease, right? Or at the minimum, they're getting some community acquired pneumonia treatment. They're getting antibiotics. How much secondary bacterial pneumonia or co-infection with bacteria is playing a role in these cases, we don't really know at this time. We don't have enough autopsies to say, and we have this issue that everybody's getting getting antibiotics. But it's possible that we are actually preventing a lot of deaths because we are treating people with antibiotics, which is preventing them from having whopping bacterial pneumonias on top of COVID. And that if we weren't doing that, maybe we would be approaching numbers similar to 1918. I don't know. Or it may be that the 1918 virus was just that much worse. Okay. In a typical bad influenza year, where we have, say, a newer version of the flu virus, it's a vaccine mismatch, or we don't have a vaccine, we see about a 0.1% mortality rate. And that mortality occurs in a U-shaped curve where the elderly, the people that we're talking about over 65, particularly ones with comorbidities, and the very young children, babies, infants, and young, preschool age children tend to be the most affected, and we have significant numbers of deaths. With this pandemic right now in the United States, the mortality rate based off the cases that have been recorded is now down to about 3.7% as of yesterday. That does not represent the full spectrum of cases in the United States. And my Serosurvey that I'm doing and others are doing will hopefully help us understand that. If we find that as few as 5% of the population that has not been diagnosed with COVID, meaning they're not recorded as cases, actually have antibodies, meaning that they were likely infected at some point and were asymptomatic or minimally symptomatic and got over it, then that would drop that mortality rate to less than 1%, around 0.7%. If that number climbs higher, more than 5% of the United States, 10% or more, now we are approaching a bad flu year in terms of mortality rate by percentage, right? So it depends on how you look at it. But at the same time, the total number of deaths may be very higher than a typical bad flu year. Because in a typical bad flu year, we have lots of people that do have some level of immunity and are protected. And so we don't have as many cases as we do during a pandemic like this. Now, in a flu pandemic, all bets are off. You could have as many cases as we do now. But in a typical bad flu year, you don't have as many cases. So it depends on how you look at it. If you look at it as a percentage or a mortality rate, I think we are going to end up somewhere close to a bad flu year. But if you look at it as total deaths, it's obviously going to be a much larger number of total deaths than just a bad flu year, and similar to a bad flu pandemic. Worse than some, better than others. The other thing I would say on this topic is that, sorry, I lost my train of thought here on this. We also don't have children dying during this pandemic in large numbers. And so again, that's another way to look at the deaths and the dying, is a 1% mortality, almost all in people over 65. Is that worse than a 0.5 or a 0.1% mortality where 25% of those are small children? I'm not saying there's a right or wrong answer to this question. I'm just saying these are things we have to take into account as we think about comparing pandemics and epidemics as what we consider worse or better. And so these are very difficult questions and things to think about.
Man, this is great. I really thank you for all the detail and insight that you're providing us. And Mary Ellen, I think because we've gone so long, I'm thinking this last question here, because we could go on all afternoon with the questions that are coming in. This last question from Susan Rannick may be a good way for us to just wrap up what has been a great presentation. And Susan Rannick says, as you watch the news reports and the stats, knowing what you know, if you were standing on the nation's podium, what would you tell the general public to do?
Wow. Well, again, that's not something I've been tasked with doing at this point in time. And so I don't want to stand in for anybody in my institute or in the government, but personally, I feel the most important thing we could do is really educate the public. And I think, and this is my personal opinion, I don't think we've done enough to truly do that, to really be open and really educate the public as to what their risks are, what the risk factors are, what the reality of masks are, what the reality of what we know about transmission is, how to use the masks if you're going to use the masks. I think we need to be clear. I think the general public is hearing a lot of messages. A lot of those messages are conflicting with each other, coming from different people at different times, sometimes from the same person at different times. And I think it's very hard for people to make good quality decisions about what they should and shouldn't do. And so personally, I don't know that I could tell you exactly what I would say specifically to people what to do, but I would certainly want to try to educate them more and clearly about what we do and don't know. And really what I tried to do with you today, and I hope I was able to do that. Again, I'm not perfect, and I'm sure there's some things that I said today are going to turn out to be completely wrong in the future. But to me, I think we have to be open and honest about what we understand and what we can learn from our previous experience with respiratory viruses, and give people as much information as we can to make decisions about how they want to handle things moving forward. And also, we can't be afraid. We can't be afraid. I showed you those numbers. Over 600,000 people die from heart attacks. Not heart attacks, I'm sorry, heart disease every year. Those of us who are, over 40, or especially those of us who are men, we are at risk of that. And so if I'm walking around scared of getting COVID, I should be really scared of having a heart attack or having some kind of heart problem, come up and happen. And so, at some point we have to put things in perspective, and we do have to live our lives. Now we have to take proper precautions, but we do have to live our lives. So I think we're really walking a fine line and we have a delicate balance of, can we do things to prevent spread and prevent deaths, but at the same time not destroy, you know, everything in our lives and be so afraid that we're not living, because, you know, we have a case fatality rate of living of 100%. We're all going to die at some point. So that's an important thing to keep in perspective.
Well, Dr. Memoli, this has been invaluable. I think in some ways I feel more reassured and less afraid. There were a few points there that made me more concerned, frankly. So, you know, if we have a 2 or 3 year thing ahead of us, but we may as well know that that is a possibility. So we really, really appreciate all that you shared with us today. And thank you for coming.
Thanks for inviting me. I hope this was helpful.
Absolutely.
Oh, that's great. Thank you. All
Sure.
right.
All right.
We'll see everybody. Okay. Bye-bye.
Bye-bye.